Okay, so I want to talk about calcium in the heart, mainly in health, but I'll have a little bit at the end about calcium in disease. If you want inspiration, the younger people in the audience, you only have to walk a few hundred meters from here into the Dom Square to see the memorial to Albert Szent-Györgyi, who's, of course, as we've heard already today, discovered and characterized vitamin C. Much more recently, of course, as you've also heard a couple of times today, Katalin Karikó is another role model for you all. So you've no shortage of role models. I thought perhaps the educationists tell me you should give learning objectives before you give any talk. I couldn't really think what the real learning objectives were, so I thought I'd set you a quiz. So what do these four things have in common? This is a limestone quarry somewhere in Hungary. I got it from the internet. This is a safety lamp designed such that coal miners could have illumination underground despite the flammable atmosphere. Here's an ECG or an EKG, and this is an intravenous drip. And I hope in 30 minutes from now, you will understand what these have in common and how it relates to my talk. So the talk's in three parts: an introduction, calcium in health, calcium in disease. I thought I'd tell you a little bit about me, but I think Andras has shown at least one of the photographs I was going to show. I was born in 1955. Thank you. And I noticed from Dr. Kobilka's talk that 1955 was obviously a good year to be born, if I got that right. It was a time before color photography was around, clearly. You've seen this picture already as a graduate student with John Lederer. He's already told you the only thing people know about Manchester is not the university, but the football team. But perhaps most importantly, I do have a life outside science. Our younger son here got married last year, and these are our 3 children, their partners, and our 5 grandchildren. Aren't they cute? So, let me introduce you to the heart. I'm sure you all know about the heart. The heart, we've heard about trillions of dollars from the previous speaker. I can't match trillions, but the heart beats 2 billion times in your life. And it's important that it does that. It pumps an awful lot of blood. In fact, the total amount of blood that your heart will pump in your life will fill 3 supertankers with blood. So we all start on the same page, here's the heart. There are 4 chambers, the 2 larger ones, the ventricles, the 2 smaller ones, the atria. The heart receives deoxygenated blood from the body. It's pumped through the right side of the heart. It goes to the lungs, where it's oxygenated, brought back to the left atrium, then the left ventricle, and then it's pumped out of the aorta into the body. Well, the heart is simply a muscle. It pumps. This is an ultrasound of a human heart, and it's roughly the same orientation here. And I think you can see that each time it beats, it pumps, and you can see the valves which are designed to make sure the blood only pumps in one direction. The reason it can pump is it's a muscle. Now, typically one thinks of muscle like skeletal muscle in one's legs and arms, but cardiac muscle is just another muscle, and the walls of the heart here are made up of muscle cells. Here's a cartoon. You get this collection of all these individual cells which form a group, a syncytium, one cell, another cell, and a third cell. These cells are electrically coupled, and there is an electrical impulse, which you can measure as the ECG, which establishes contraction. I'm not going to talk today about the electricity in the heart. What I want to focus on is what comes next. Now, yes, science is properly interested in the heart, but people have been fascinated by the heart for hundreds, if not thousands of years before modern science. And I hope the reason for that is obvious. All you have to do is put your hand on your chest or on a vessel, and you can feel the effects of your heart pumping. My colleagues who work on things like the kidney and the brain can't identify what their organs of choice are doing. But for the heart, we can do that. And therefore, it's not surprising that the heart has inspired artists, authors, and musicians for many years. I thought I'd find a Hungarian example of music and the heart. And you've got, obviously, very distinguished composers, such as Bartók, in this country. I couldn't find anybody who'd worked written about the heart or composed about the heart. So I'm going to do the next best thing. Schubert, although he lived in Vienna, that was obviously the time of the Austro-Hungarian Empire, so he's an honorary Hungarian as far as I'm concerned. So, in the best sense of collaborative science, Schubert, the composer, collaborated with Müller, the poet. And I'm going to play you a little bit from a song cycle called The Winterreise, A Winter's Journey. And the person who's singing is waiting for a letter from his loved one. And understandably, therefore, he's rather agitated. And I want you to notice two things. One, in a moment, the piano accompaniment, which I think is a wonderful rendition of an agitated heartbeat. But the other thing is this sentence in the paper, which is really which really describes what I've been interested in for my scientific career. Was drängst du denn so wunderlich mein Herz? Why do you beat so mysteriously, my heart? And my Hungarian colleagues assure me that this is a correct translation, so I'm hoping. So just listen to this. I think you'll have to agree that that piano accompaniment really sums up a heartbeat. So that's Schubert's take on the heart. Let's turn more seriously now to science and the heart. If we're interested in calcium in the heart, it all starts off with this man. This is Sidney Ringer. Ringer was a physician at University College Hospital, London in the 1880s, and he saw patients. But every day before he saw patients, he carried out research. And what he was interested in was the fact it was already known that you could take the heart out of an animal and it would continue to beat. Now, the fact that we can take the heart out of someone is, of course, essential for cardiac transplantation. You take the heart out of a donor, and in the absence of any nerves, the heart will continue to beat in the recipient. And he wanted to know what was required for the isolated heart to beat. And in 1882, he published a paper saying that all you need for the isolated heart to beat is a simple solution of sodium chloride. Now, if that result had stood the test of time, I wouldn't be here today talking to you about my research, and Professor Varro's research would also have been less bright. Fortunately for us, a year later, he re-evaluated the situation. He published a paper in the Journal of Physiology, which Andras mentioned, and this is the title of the paper: A Further Contribution Regarding the Influence of the Different Constituents of the Blood on the Contraction of the Heart. Now, a word first to the younger people in the audience. When you publish your first paper, if the first three words of your title are a further contribution, your paper will probably be rejected immediately, because in today's trendy age, a further contribution sounds like it's not novel enough. But fortunately for Ringer and for us, the paper was published, and the most famous sentence in the whole paper is this one: I discovered that the saline solution which I had used had not been prepared with distilled water, but with pipe water supplied by the New River Water Company. So distilled water is, of course, highly purified water. That's what he'd meant to use, but instead he'd taken pipe water, water from the tap or faucet, and used that. Once he realized the mistake, he immediately repeated the experiment with sodium chloride in distilled water and discovered that the heart stopped beating immediately. And Ringer therefore correctly hypothesized that the reason that the heart beat in pipe water, but not in distilled water, was because one of the contaminants in pipe water was required for the heart to contract. He commissioned an analysis of the pipe water and added back the contaminants one by one. But only when he added back the calcium chloride did the heart start beating again. So Ringer therefore correctly concluded a year later in 1883 that calcium was required for the heart to beat. And we know now that calcium is required for the normal function of essentially every tissue and organ in our bodies. That's the scientific story of Ringer, which some of you may know, but I want to share with you two other facets of the Ringer story. One is that the clinical world owes Ringer an enormous debt. You take it for granted that in hospitals in the world, we can safely give patients extracellular fluids from drips like this. But have you ever thought about the fact that this depends upon the fact that we know what to put in these extracellular fluids to keep the patients and their organs healthy? And the fact that we can do that depends upon the work of Ringer and others. The other message I want to share with you, I'm afraid, is a slight digression into English grammar. If you look at this sentence here, look at the grammar of it. I discovered, here's the translation, active voice, that the saline solution which I had used, again, active voice, had not been prepared, passive, with distilled water, ... In other words, I think some of you have got it already, what Ringer is saying is that he was the clever guy who made the discovery, he was the hard-working person who did the experiment, but somebody else in the passive voice was at fault. So I hope it never happens to you in your bright scientific careers, but if you ever have to, 1 year later, publish the reverse result from what you published the year before, decide now whether you're going to be a generous person who takes the blame yourself, or perhaps, like Sidney Ringer, implicitly blame somebody else. That's the Ringer story. Even today, we know the isolated heartbeats. This is a rat heart being superfused, and it carries on beating. And as I've said, this is essential for cardiac transplantation. In many of our experiments, we don't study the whole heart. It's more convenient, and you can do more detailed analysis if instead you study the isolated cell. I showed you the cells in my cartoon a few slides ago. This is a ventricular myocyte. It's about 100 microns, 0.1 of a millimeter long, and each time it's stimulated with electricity through its pipette, as happens during the normal heartbeat, it contracts. The cell shortens. And if the billions of cells in the ventricle all shorten, the ventricular volume decreases, the pressure inside the ventricle increases, and eventually blood is ejected out of the ventricle into the aorta. So this is what's happening in all the cells of your ventricle as you sit here now. But what's the origin of this contraction? Well, it's been thought for many years that this contraction depends upon calcium, and it was suggested that this could be because intracellular calcium rises and activates contraction. And indirect evidence in favor of that was the fact that if you inject calcium into either a cardiac muscle cell or originally a skeletal muscle cell, the cells contract. So calcium is capable of causing contraction, but that doesn't prove that calcium rises. And if you really want to know whether calcium changes during this contraction, you need a way to measure calcium. A slight digression, if you think of advances in science, to simplify it slightly, they come in two sorts. Some of them are conceptual advances. Think of the theory of relativity. But others are questions that have been around in the literature for quite some considerable time, but the techniques haven't been available to measure them and to study them. And in order to study calcium in the heart, the field owes an enormous debt to another Nobel laureate, who is Roger Chen, who got the Nobel Prize in chemistry in 2008. And what Roger did was to produce, amongst his many accomplishments, was a chemical which sensed intracellular calcium. So this is one of the chemicals. These negative charges at the top are the region that calcium binds to. I hope there are no real chemists in the audience. So calcium binds to these negative charges at the top. This part of the molecule here fluoresces. So if you shine a short wavelength light at it, it emits a longer wavelength light of a different color. And the important thing about this molecule is the amount of light it emits, as shown here, now as a function of wavelength, depends upon the calcium concentration. So at low calcium, you get very little light emitted at this wavelength. As you raise the calcium over the physiologically relevant range, calcium goes up. So Roger Chen has provided the field with chemicals which can sense calcium concentration inside cells. So now it's easy to take a cardiac myocyte like this, a ventricular myocyte, load it with one of his indicators, and measure the amount of light emitted. So each time we stimulate it, what I want you to see is that the color of the cytoplasm, the body of the cell, changes, and this indicates a rise of intracellular calcium. And it's this rise of intracellular calcium that activates contraction. You may notice that these cardiac cells have two nuclei, and calcium in the nucleus changes somewhat more slowly than that in the cytoplasm, but that's not really for today's lecture. We can quantify calcium, as shown down here. At rest or in diastole, calcium has a concentration of 0.1 micromolar, or 100 nanomolar, and it rises to about 1 micromolar during contraction, or as we call it, during systole. And it's changes of the size of this calcium transient which are responsible for changing contraction in the heart. So when we exercise or run, our hearts beat more strongly, and that's because the rise of calcium gets bigger. When people have heart failure, their hearts beat more weakly, largely because this rise of calcium is less. So this is measurements of calcium inside the cell. Where does the calcium come from? Here's a cartoon of a cardiac cell. This is the extracellular fluid. This is the intracellular fluid, called the cytosol. The cells have a large diameter, and they have these invaginations into them called transverse tubules. There are two sources of calcium. One is calcium enters the cell from outside through these channels like this, the so-called L-type calcium channel, which when it opens allows calcium to enter. But the main source of calcium is calcium being released from this intracellular store, the sarcoplasmic reticulum, which for the rest of the lecture I'm just going to call SR. So two sources of calcium. So during your, and the other thing I should point out, these are the contractile proteins in the cell that slide over each other to make the cell shorten and produce force. So during your heartbeat, first calcium enters from outside. This makes these channels here open to release much more calcium. So we get the rise of calcium, as I've shown you. This calcium binds to the protein troponin on the muscle filaments here. Once it's bound to that, this allows the filaments to slide over each other and the cell to shorten. And this is an energy-requiring process. If the heart's going to work as a pump, not only must it contract, it must also relax. And relaxation requires that calcium be lowered to resting levels, partly by being taken back up into the SR and partly by being pumped out of the cell. So that's where calcium comes from and goes to during the cardiac calcium transient. So as I've said already, the force of contraction of the heart and the calcium transient vary during normal life and in disease. So which of these many steps controls it? I'll spare you most of the evidence, but there are two things that control it. One is the size of this L-type calcium channel, how much enters, and the other one, perhaps the most important one, is the amount of calcium stored in the sarcoplasmic reticulum. As I'll show you in a moment, as you increase this, you increase the size of the calcium transient. I'll give you one example of that, and this comes back to what Brian Kobilka was showing us before. So these are on a slow time scale. Every time we stimulate the cell, calcium rises, and I'm not showing you the cell contracts. And if it was in the heart, it would be pumping blood. This is under basal conditions. Now we add the hormone adrenaline or epinephrine. In fact, we're adding a synthetic version of it, isoprenaline. And this leads to what he called the fear and flight response. If we're frightened or want to be aggressive, we all know that amongst many things, our hearts beat faster and more strongly. And the reason they beat more strongly is that during fear and flight, adrenaline is released. As we've heard, it binds to a G protein coupled receptor, and this results by effects on the calcium current and the amount of calcium in the SR into an increase of the calcium transient as shown here. So the calcium transient goes from this size here to this size here. So this is what's happening when you exercise. Adrenaline makes the calcium transient larger. I told you that we have two sources of calcium, so you might say to me, how do I know where this calcium is coming from? The way you can do that is not just have a calcium-sensitive indicator in the cytoplasm to measure these calcium transients, but as well have a different calcium indicator emitting at a different wavelength in the sarcoplasmic reticulum. So you can measure the amount of calcium in the sarcoplasmic reticulum at the same time as that in the cytoplasm, and that's shown here. So under basal conditions, this is cytosolic calcium. It goes up on each heartbeat. This is the amount of calcium in the sarcoplasmic reticulum, and you can see it goes down on each heartbeat before recovering. And that's because a large fraction of this calcium here is due to calcium released from the sarcoplasmic reticulum. When we mimic the fear and flight response by adding the version of adrenaline, epinephrine, then you can see the calcium transients again get bigger, and that's largely because the amount of calcium released from the sarcoplasmic reticulum also gets bigger. So direct evidence that calcium released from the SR is important here. And in other experiments we've done, we've quantified this. We've measured the size of the cytosolic calcium transient and experimentally changed the amount of calcium in the sarcoplasmic reticulum, and you get this incredibly steep relationship between the amount of size of the calcium transient and the amount of calcium in the SR. And we think this underlies many of the conditions in which the force of contraction of the heart is increased. Finally then, let me turn to calcium in disease. Let me give you a very brief introduction, and now it's my time to hope that there's nobody with a medical degree present in the audience, which may be an even harder problem. The heart is a muscle. As a muscle, it requires an oxygen supply. If it's deprived of its oxygen supply, the heart dies. Oxygen is carried to the heart through the coronary arteries that carry blood in the walls of the heart, in this case, and the ventricle. This is what a normal coronary artery looks like. Large diameter, very smooth walls. If, however, you have atherosclerosis, the coronary arteries don't stay open. There are regions like this here where they're narrowed. And atherosclerosis decreases the oxygen supply to the heart. This is a post-mortem slide of somebody with atherosclerosis. Now you can see the lumen, the inside of the vessel, is reduced to this size. This dashed line here shows you what the original size of the coronary artery would have been, and the rest is made up by this fatty, made of lipid core with a fibrous cap. This is bad enough. This will grossly decrease oxygen supply to this part of the heart, will cause pain known as angina. But what quite often happens is that the cap breaks down, bits fly off and completely block it, or there's a blood clot which blocks it, and no blood gets to that region of the heart, and part of the heart will die. If enough of the heart dies, then the patient may drop down dead immediately, but if only part of the heart dies, you get this phenomenon here. So in this cartoon here, this part of the heart is dead. We say it's infarcted, perhaps because its blood supply was stopped. The rest of the heart can compensate. It has to work harder to compensate for the fact that this part isn't contracting. Because it's working harder than it was designed to do, experience shows it will eventually fail, and you get a condition known as heart failure, where the heart eventually can't pump enough blood to satisfy the needs of the body. And two things happen in heart failure. The first one is, as I've said, you don't pump enough blood for the needs of the body. So the question, and I'll come to the other one in a moment, the question is, why don't you pump enough heart? This is from a study we did several years ago. These were experimental heart failure induced in ferrets, and these are calcium transients from normal ventricular myocytes, and these are typical ones from ferrets which had undergone heart failure. And you can see a much smaller calcium transient here than here, leading to a reduction of contraction. So one of the things that happens in heart failure is that the rise of calcium is smaller. The other thing that happens in heart failure and is responsible for the death perhaps of half the people who have heart failure, is not a primary reduction of contraction, but rather the genesis of arrhythmias. Normally, I don't know if anybody here has ever had an ECG, an EKG, you get a signal that looks like this. The ventricle, the electrical signal of the ventricle is this large spike here. This is what in a normal person. If you have a catastrophic arrhythmia, rather than having an ECG like that, you have an ECG like this, and this is called ventricular fibrillation. If you look at the heart that's fibrillating, rather than contracting and relaxing roughly once a second, it will just be shimmering. It'll be contracting in a completely discoordinated way, and it won't be pumping any blood into the rest of the body, and therefore once it stops pumping blood, the brain will very soon die due to lack of blood, and indeed the heart itself won't be getting its blood supply. So arrhythmias, ventricular fibrillation is lethal. Well, you might be asking, what's the link between ventricular fibrillation and calcium? I showed you before a smaller version of this, and I told you that on each heartbeat, calcium enters and calcium is released from the sarcoplasmic reticulum. That's how it's designed to work. Under some conditions, however, calcium leaks spontaneously out of the sarcoplasmic reticulum, and it can do this at any stage of the cardiac cycle. So what happens then is that calcium leaks out, some of it is pumped back, some of it is pumped out, but if enough leaks out, it will go ahead to the next region of the sarcoplasmic reticulum, and it will make these release channels open, releasing more calcium, and so on and so on. And what you predict then is you get a wave of calcium release propagating down the cell. Now, in the original video I showed you, I made the point that calcium release is uniform throughout the cell, so that contraction is uniform. Under these conditions, it's not uniform. What you get is calcium released here at this end of the cell, and it propagates down. So that means that the contraction in the cell is out of phase, is discoordinated in the various regions of the cell. It's out of phase in the different cells in the heart, and as a result of that, this heart will contract much more weakly. It's worse than that, however. I don't have time to go into detail, but these abnormal changes of calcium, which can happen at any time in the cardiac cycle, affect membrane currents in the surface membrane and produce abnormal electrical activity, and that's what kills people. So just as normal calcium cycling is essential for contraction, abnormal calcium cycling leads to arrhythmias. And we've shown that the probability, the frequency that you get these calcium waves is a steep function of the amount of calcium in the sarcoplasmic reticulum. And the method we used to measure SR calcium content was devised by Andras. So below a certain threshold level, you never see the calcium waves. That's the healthy condition. Above this level, you get calcium waves. So raising the calcium in the SR produces waves. We also know, however, that there are people who are born with abnormal release channels in their sarcoplasmic reticulum, and for them, the threshold to get these waves is much less, so they're more likely to get them. And also that in heart failure, the release channels are more likely to release and produce waves, possibly because the channels are more phosphorylated. Whatever the explanation, if you get these calcium waves above a certain level. Right, let's end at the beginning of my talk. What I hope you realize these things all have in common is calcium. Limestone, of course, the major metal in it is calcium. The element calcium, I didn't tell you, was discovered by the chemist Humphrey Davy, who also developed this so-called Davy lamp so that people could work safely in flammable atmospheres. I've told you how calcium is related to normal and abnormal cardiac function, and I've also told you how Ringer is so vital to medicine. Finally, I just want to congratulate the students on this program and equally their dedicated teachers, and to all of you with what the world can throw at us, good luck. Thank you very much.
Thank you very much, David, for this excellent presentation. I'd like to ask the students whether they have any questions for Professor Eisner. Yes, there's one. Do we have a microphone, maybe, Hanny? I think the microphone is not on. Maybe it needs to be turned on.
Is it on? Yeah, okay, great. So I've really enjoyed the presentation. And my question would be, you mentioned that during the life of a cell, it needs calcium to be able to contract. And why would calcium flow into the cell? And why would it release calcium? What makes the cell do this?
What makes
the cell release calcium and increase its calcium level?
What makes it? Well, during the normal heartbeat, you get, first of all, an action potential. The voltage across the surface membrane changes, and that opens the calcium channels in the surface membrane. Why is calcium released from the sarcoplasmic reticulum? In cardiac muscle, that's a phenomenon called calcium-induced calcium release, where the calcium entering from outside binds to the release channel, changes its shape, makes it open, and releases calcium. In skeletal muscle, the process of release is different, but that's what happens in the heart.
Thank you very much.
Thank you.
Any other questions from the audience? Yeah. No more questions? There's one in the back.
So, if these arrhythmias are caused by the irregular calcium intake, can it be cured? Can these arrhythmic cells be cured?
That's a great question. So, one way you might think of curing them is to stop the calcium release from the sarcoplasmic reticulum, because that will stop the waves. The problem is it will also stop the normal release, which you rely upon for the heartbeat. But there are drugs that people have used, and flecainide is one of them, which seems to interrupt the abnormal calcium release more than the normal release, and there is hope there. So there's a group of people who have an inherited condition called catecholaminergic polymorphic ventricular tachycardia, which I think the next speaker may refer to. And they're born with calcium release channels, which are much more likely to release calcium. In the past, they were given beta blockers, which have side effects. Now they're more likely to be given these drugs, which seem to block abnormal calcium release.
Thank you very much.
So, if there are no further questions, we would like to hand over. Do you want a question? Oh, sorry. Yes. 3 rows to the back.
Next year, you need to invest in a second microphone.
I think lecture hall is a bit too big.
First, I want to thank you for this opportunity that I can ask you a question. I know an unfortunate condition which people are born with when they born with a bigger heart than normal people, but I don't know why they have a shortened lifespan.
That's an interesting, difficult question. So you're talking about hypertrophic cardiomyopathy. I think. Sorry, that wasn't supposed to be a joke, but still. So these people have a larger heart, and there are many causes of that. An interesting one is, for many of them, it's a genetic cause where the muscle proteins have a mutation, and that leads to irregularities of contraction. there are various theories for why it happens. One of them is that because they tend to contract more, then it gives them energetic problems. They run out of ATP. But another explanation is that their muscle proteins bind calcium more strongly, and that interferes with normal calcium cycling. But it really isn't worked out yet. And if you're looking for a research project for the future, that would be a great one.
Thank you very much.
There's one further question in the back.
I would like to ask what happens if that the calcium level in the heart reaches a very high level in the heart for a very long period in time.
What happens if the calcium reaches a very high level?
Yes,
Is that what
in
you're
the
asking?
heart for a longer time period.
Well, so that can happen following ischemia. So I talked about ischemia, where the heart is deprived of its oxygen supply. If that stays for long enough, the heart cells will die, partly because calcium reaches a very high level, affects mitochondria, and activates various enzymes. But what's also interesting, and there are people here who work on it, is if you have briefer ischemia, when you relieve the ischemia, as you do when somebody comes in with a blocked coronary artery, you open up their coronary arteries, that reperfusion can also lead to damage. And again, that seems to be associated with a large rise of calcium. So the heart is designed to have brief rises of calcium on each beat. It's not designed to have a very long-lasting rise of calcium. Thank you.
Thank you very much.
I think there's one last question in the very back.
In your presentation, you showed a graph which depicted how calcium waves are triggered by excessive amounts of calcium in the heart. So does that mean that by, say, hyper-regulating calcium levels in blood, we could prevent these potentially fatal heart abnormalities?
I think the problem is that if you change the calcium level in the blood, you'll do terrible things to the function of nerves and skeletal muscles. Nice idea. I don't think that would work.
Okay, thank you.
But thank you.
Thank you very much. Unfortunately, there's no more time for questions behind them time. We'd like to hand you, David, a certificate of appreciation, and thanks again for coming to Szeged.